Hi everyone, this is a tutorial about meiosis or sexual reproduction for those who have not gotten to it in lecture yet. Sexual reproduction is defined as the union of two germ cells or gametes, one from each parent, whose cells then combine to form a zygote. These specialized cells are either an ovum or egg or spermatozoan or sperm. Eggs and sperm each have half the number of chromosomes that you would find in a normal body cell of that individual. If gametes were formed by mitosis, the way that body cells are formed, they would each have a full set of chromosomes, and then combining them would lead to offspring with double the chromosomes of their parents. The chromosome number would then continue to double in each successive generation. Which obviously is a problem. You then have exponentially more and more number of chromosomes. But then if prior to fertilization maybe one parent's chromosomes were eliminated to keep that number the same, you would have the correct number of chromosomes, but you would eliminate the genetic contribution from one parent over the other. So in order to get a consistent number of chromosomes, and for each parent to contribute to the next generation, the gametes need to efficiently, accurately, and safely reduce their number of chromosomes. This is achieved through meiosis. So meiosis is a mode of cell division in which each cell has only one of what was a pair of chromosomes. Remember that we humans have 23 pairs of chromosomes, or 46 chromosomes in all. One chromosome set from mom and one from dad. Gametes need to reduce each pair by exactly half, but maintaining one chromosome of each set because you want to have a code for each thing that you need to survive. So this is done by replicating each homologous chromatid once, but dividing them twice. So homologous chromosome is one chromosome from each of those 23 pairs. So each chromosome duplicates, lines up with its pair, and then divides twice. So first the homologs will separate each pair, which occurs right here. And then in meiosis 2, the second division, sister chromosomes separate, so the two copies. If we use humans as an example, again, we begin a normal body cell as a diploid. It has two sets of 23 chromosomes or 46 chromosomes. Then in prophase, each chromosome duplicates itself. This creates a double diploid, a cell with 92 chromosomes. The homologs separate in meiosis 1, so these are homologs. They code for the same thing. And then that creates two diploid cells, still 46 chromosomes. And then a further division in meiosis 2 allows the sister chromosomes to separate, and the result is 4 different daughter cells, each having half the number of chromosomes that the original parent cell had. So in this case from 46 to 23 down here. The second meiotic division, which you can see in this area here, meiosis 2, is similar to mitosis because sister chromosomes are separating, or sister chromatids technically. However, these chromatids aren't identical as they are in mitosis due to this process of crossing over. So here's a diagram of crossing over, which before the first division, so in prophase 1, the homologous chromosomes from one parent exchange corresponding segments with the other parent. Recombination, in this case, allows the 4 chromatids that result in the end down here to contain a different combination of genes from each previous generation. So it combines both grandparents' genes for within the parent for their offspring. So the result of this is that each gamete has a unique combination of traits from each parent, which results in what we can consider a seemingly infinite number of genetic combinations when the two zygotes form. Well, when the two gametes form into a zygote. Even if two parents reproduce constantly, as we can see in some large families like the Duggars who have 19 children, no two children born of separate reproductive events will be exactly alike. So unless they're identical twins and came from the same egg and sperm combination, they will always be slightly different. So now on to non-disjunction. Non-disjunction is the failure of chromosomes to separate during anaphase of either meiosis 1 or meiosis 2. An uneven distribution of chromosomes in the daughter cells is a result of this. One will have 2 chromatids from the parent and the other will have none. Both situations can have equally disastrous genetic effects, including non-viable cells, so cells that don't result in a living being, or disrupted genetic development, such as Down syndrome. So Down syndrome, you can see here, it's the most well-known trisomy and is caused by non-disjunction on the 21st chromosome. The result is that a fertilized egg will have both copies of one parent's 21st gene and one copy from the other parent, creating 3 copies, which is why it's called a trisomy, where there should only be 2, as you see in all the others. So a sex-linked gene, which you'll see sometimes, is a trait that's coded for on the 23rd pair, which is this pair down here. This pair determines the sex of an individual. An XX combination is for females and an XY for males. A male normally inherits their X from their mother and their Y chromosome from their father, which is why you can trace sex-linked genes through generations depending on whether the offspring was male or female. The inheritance of the traits linked to the X chromosome are calculated using a Punnett square that looks like this. Because the Y chromosome actually codes for different traits. It codes for traits specific to being a male. It's not homologous to the X chromosome that sits next to it in the 23rd pair. And because it doesn't have homologous genes, a male will express only the gene that they're given by the mother's X chromosome. So when you do a sex-linked Punnett square, your genes only go as an exponent to the X. That gene is not carried on the Y, so it cannot contribute in this case. So here you see that the male is expressing only the trait that it was given by its mother, and here the other X that it was given to by its mother. So the mother has 2 possible X's, but the male will express whatever it is given to, whichever X it is given. So oogenesis and spermatogenesis are not the same. So oogenesis is the production of eggs, spermatogenesis the production of sperm. And they differ in 2 main ways. First, when they occur in the individual's life, and second, how many usable cells are a result. So spermatogenesis begins in a male life starting at puberty and ending at death, whereas oogenesis begins much earlier. A female is born with all of the eggs that she'll ever have, having been produced in the first month of her fertilization. So while she is still in the womb. The process of making fully formed gametes usually begins prior to a girl's birth, but the egg won't be finalized until after the girl begins to menstruate, so it won't become a true gamete. All gamete production ceases at menopause, but each month after sexual maturity, one of the eggs in this stage is selected for and released. So it finishes maturation and is released. Meiosis in a male, however, produces 4 active sperm cells. Each are working, while meiosis in a female produces 4 haploid cells, but only 1 actual usable cell. The other 3 cells are called polar bodies. They are unused, and then they become reabsorbed into the body. Okay. So finally, one of the last things that you'll look at in lab is the idea of genotype versus phenotype. So remember, if you don't know the difference, a genotype is the actual genetic material or genes present in the cell, whereas the phenotype is the physical expression of those genes. So here I have an example. I have 2 purple flowers. Both are phenotypically purple, but one has 2 purple alleles. It's homozygous dominant. And the other has a purple allele and a recessive white allele. It's heterozygous. So that's the difference between genotype and phenotype. So I hope you found this helpful. Good luck in lab. And let me know if you have any questions.